All right. Hello and welcome, everyone. Today, we are going live with my special guest, Matthew Gates. Matthew is an integrated pest management specialist and principal IPM consultant for Avitas Global. We'll be sharing a presentation on pest and disease prevention and taking your questions in the chat. Matthew, welcome.
Thanks for having me.
Great to have you here. So, you've prepared quite a presentation, and I am excited to dig into it. So, I will let you take it away. What are
Okay,
we
thank you
gonna
very much.
what are we gonna be looking at first here?
Does it look good?
Yeah.
All right, thank you. So thanks for having me, Michelle. This will be a presentation about something very special to me, which is this sort of strategic outlook of how home growers and really anyone, it's echelon agnostic. So people of all scales can use this, but specifically I wanted to emphasize some aspects, some basics and fundamentals of IPM, like how plant defense works, how cannabis interacts with pests, and some of the pests we deal with and what their capabilities are, which allows us to sort of eco-max our home grows. So the title is called Everswarm Strategy, Eco-maxing home grows for future resilience. A little bit about myself. Again, my name is Matthew Gates. I'm the founder of Zentinol Consulting. I've been 16 years in the IPM support work. You can check me out at zentinol.com, and also I have some socials here. And I have some publications in various places with regards to pests or pathogens for cannabis. And if you haven't checked it out, you should look up this New York State Cannabis Sativa Guidebook. It's a collaboration with researchers from Cornell and some other people in the sort of university space working to do research on cannabis. So it's in English and Spanish. And you can check it out at your leisure. So this is a little bit of an itinerary. Interspersed, we're going to be talking about two main things. We're going to talk about how pests and their development informs IPM strategy. And then we're going to talk about how those biological aspects and environmental aspects influence cannabis, its development, and also with various pests. We'll even briefly go over a little bit about cannabis evolution, and I have some emphasis on Midwestern USA, since we have some cool research that talks about some adaptations that are happening, and also some microbiome information. So I look forward to talking about that a lot. So the everswarm as a conceptual model is this concept of a sort of constantly evolving pest population. There are many different species that we contend with. There's fungi and bacteria, there are various insects and mites, but there's also enabling factors, microbes inside them and around them, and even in the plant that can help them, as well as the environment. We won't get into all these examples here, but one of them we will, which is the budworm moth. It's an insect, yes, but it has not only its own capabilities, but those of bacteria in its gut that allow it, like Enterococcus does, to destroy things like Bt proteins that are produced by Bt, which is a type of bacteria commonly used against budworms. It's one of the only options people have had in cannabis for them that's safe and effective and not a toxic chemical. And I've said here that basically you can't be quicker than a surprise ambush. So when pests come around and you don't have a plan for them, there's no way you're going to do better than them. Essentially, it's very difficult to do this. And definitely, this is the case if they're overwhelming, which is often the case when they come in, especially in outdoor and greenhouse crops. So biosecurity can be a way to handle that. But they're also sort of genetically honed to bypass all the different selection pressures they've learned about as populations and evolved for in other parts of the world. So even if you grow in a responsible way, it doesn't necessarily mean other people are, and other crops certainly have their own contingencies, and this influences us. So one pest is simple. It's like a Bruce Lee flick. One guy honorably comes out, you kill him. He comes out again, you kill him. But 40 plus at the same time, or 10 at the same time, or 4 at the same time is way more complex and difficult. I'd also encourage people to review and analyze educational content, trust but verify all sources and claims, including the ones that I'm making here. I definitely encourage people to check out the research, and I'm going to be going over some diagrams here that need more than the seconds I'm using here to make a point to digest fully, and I encourage people to come back or contact me if you'd like to know more. And I would also say that don't roll over for no data. Anecdotes are not data. New information is constant and complex, and so this is an example of a collaboration where I'm trying to get science communication out, but at the same time, always just be looking for sources for that information. So here would be one of those examples. This diagram probably looks a little bit complex, but it's essentially here to bring up a point, which is that if you live in places like North America or Southern South America or Australia, or many of the parts of Europe, these light blue sections are places that are highly likely to get invasive pests because of trade and because of a lot of other things, mostly agricultural factors. It's the number one thing that causes pests to move from one place to another. Now, the cost of these invasions are not always going to be the same, as we can see in section B here, but iterated this here for people depending on where you're from. You might find this to be useful for a sort of a rough gauge of whether you're likely to find new pests or not in the future. One Connex box was found famously in the UK, starting and ending. Over 421 days, it moved 75,000 kilometers by ship, about 5,000 by train, and about 2,000 by road. By road. It went all up and down Asia. It went through the Strait of Hormuz, Strait of Gibraltar, all through USA. These pests oftentimes move on these freeways. Container freight and air freight has been going up quite a bit since the early 90s and is expected to continue even with all the things going on right now. I think this is still true. And trade deals and agreements have also increased very rapidly, hockey sticked also in the early 90s. So alien species records and imports as a percent of GDP are positively correlated, and they're strongly correlated. As imports increase, the likelihood of these other organisms, some of which might be more or less benign, although it's hard to track these things a lot of the time, others can be crippling pests in agriculture and in cannabis for our purposes. So I would say that strategic IPM, it has to be locally acceptable, it has to be sustainable, it has to be varied to deal with all the different kinds of pests you may encounter and also their subpopulations that may emerge. It has to be rapidly deployable. And I would also say it should be, I said this already, but kind of communally, locally suitable, but communally acceptable. This creates sort of a layered resistance. One of my examples for this would be the two-spot spider mite, which I've nicknamed the plant poison drinker. It's got a truly egregious host plant count of about 1,000 plus, which is insane. Some records I know are 4,000 plus, which would make it the most, basically the most specious host range of any herbivore ever.
Matthew, is this the common spider mite that we find on cannabis?
Yes, sir.
Yep.
So we find that over 500 resistance genes have been pegged related to the resistance of over 96 pesticides, which is also an incredible amount. So spider mite chemical resistance, it overlaps with plant defenses. So just because they resist these noxious chemicals, which by the way, you shouldn't be using in cannabis anyways, that's still kind of working like an exercise gym. And we have this sort of Galapagos Islands situation where the spider mites intersperse, they go to other places, they bulk up, and then they are traded to other places, right? So this research was interesting because it showed that based on their biodiversity, the spider mites, Europe is a potentially likely origin point. So if you live in this area or you perhaps get sources of seeds and plants from this area, it's possible that these populations have co-evolved more. We also know that cannabis is from the Eurasian continent, and so there's influences there that might be ancient. And in Asia in particular, there's major chemical resistance genes. This graph here is somewhat complicated, but it's actually intuitive, and there's only a few points that I think are really important to consider. So basically this shows the relationship of different samples, and you can see the different countries of origin are annotated here. On the very left, we have RO, which means Romania. They found that major parts of the genome, which makes up all the genes in the spider mite, were from other populations of species. So in this case, the Turkestani species. So these hybrids are possible and they're active, and they're allowing spider mites that we know, the two-spot spider mite, the common one, is sort of using that and becoming stronger this way. Also, Italy, Greece, France, and England were considered the base in the European region. So again, if you're in this area or you have plants that are influenced from this area, it might have some breeding or sort of growing implications. And again, the highly chemical resistant relative to all the other ones that also have resistances was found in East Asia. So it's also been found, and we're not going to read over all this paragraph, but the highlights here on the right um, that I've noted here talk about how there is this high toxin resistance. And again, this is also going to affect their ability to counter the natural defenses that breeders and I would say most growers rely on and should rely on in the future through breeding measures and things like this. But the spider mites and other pests are getting a head start. So there's implications for this as you try to plan for an IPM scenario. You might not have options in the past, natural or otherwise, to rely on. And there are resistances for things like abamectin, carbamates, and organophosphates. Many of these are banned in the USA and certainly for cannabis use. But it's incredible that they're able to do this, particularly because spider mites have one of the smallest genomes of any animal. Russet mites are smaller. But the record holder was the two-spot spider mite for a long time. It's got a much smaller, about like 1/10 the megabase pairs or nucleotides like ATGC that you might be familiar with through biology learning. So these megabase pairs, the cannabis has 900, but spider mites only have 90, more or less. So it's incredible that they're able to sort of adapt and overmatch the plant, despite the fact that they are sort of genetically less resourced. But greenhouse and agriculture transport is the basically highest reason why the spider mites are moving the way that they are. So at a macro level, we can ask for our countries to be more um, vigilant and things like this, but at a micro level, we have to be more vigilant ourselves as a group. Budworms are also something that I get a lot of questions about, and it's also sort of a mega pest because it has strong chemical resistance. It's even been able to resist DDT of all things, BT bacteria, like I mentioned earlier, and even cold environments as low as minus 20 degrees Celsius, which is insane. And this is mostly found in the Xinjiang region of the People's Republic of China. So these populations could become more prominent, and they might move into places like the US, where usually growers who are maybe above the 42nd parallel and can expect a winter to destroy the populations that go down into the soil to pupate. That might not be true anymore, which will have huge implications not just for cannabis but other crops. This is from my Budworm video on my YouTube channel, so if this is interesting to you, you can check out for more information. I go over the evolution of the budworm and other implications like this, but we can see 4 populations that are American and 2 populations, one from the People's Republic of China and one from Brazil. And basically, this is supposed to illustrate the actual locations where some of these resistances were found, or in the case in 2005, a hybridization event between the corn earworm, Helicobacter zea, which is a very common budworm that affects cannabis, and Helicobacter armigera, which is the old world cotton bollworm, which is found mostly in Eurasia, but has also been recently found in the USA and threatens like $78 billion in agricultural loss, which is incredible. So we have these BT and cold resistance genes coming from China, and we have other resistances like to pyrethroids and other natural chemistries that will probably affect us as growers in cannabis. So it's something to watch out for. You might have to consider other alternatives like physical barriers and things like this. But just to drive the point really home, this is a spread of genes in the different genomes of the different populations from Africa, South America, Oceania, Xinjiang, as you can see here, the Uyghur in the sort of russet color at the bottom right here. These are all regional differences in adaptation. But a smoking hot 18,455 genes were found to be associated with two main things: toxin processing, so plant defense resistance, and plant sugar processing, which allows them to resist cold, because the blood sugar of the insect, of budworms, is trehalose, and trehalose does not freeze uh, at freezing temperatures typically. It doesn't turn frosty. So normally that would kill the budworm, but because they have this storage in their butt and their intestinal tract in their body, it allows them to actually survive the winter. This is also a graphical spread. On the left, we have a grid where on the right of the grid we have genes. Basically, these are just the names of different genes. And on the bottom, we have chemistries, which I've annotated in green here. You can come back to this one later, but the broad strokes is that in North China and Xinjiang, there are different uses of pesticides, tons of them, 70,000 tons in an area of 10 million hectares in northern China, whereas Xinjiang has 18 tons, 6 million hectares. So there's a big difference in resistance, and we find that the North Chinese populations have these massive, higher expression, light blue section in the grid reactions to many of these chemistries. And this is going to spill over into other places. Well, it already has, and so I think it'll become more prominent in the next 5 to 10 years, and I think people should prepare for this. This is also an example, a bunch of genetic information for those who are able to discern it. But on the right here, we can see an example of where just a little small change in the production of a protein allows for the storage of this trehalose. This TRET1 mutation is what allows budworms to do this. And this is a example of the cell wall, the gut lining of a budworm. And a small change in the cell wall architecture allows it to basically not get broken down by the Bt bacteria that we usually use it for. So normally it would rip apart their gut and give them sepsis and they would die. Very effective. But now this is less capable. And there's actually dozens of these Bt resistances that might combine in various populations. If you live in North America, I like to call this sort of northeastern region the pest pocket. I'm from California, and this West Coast area also is lighter, as you can see in this diagram, but there's a lot of establishment of pests in this region. We're going to go over a little bit about suitability of environment for cannabis and how it might have some implications, especially for greenhouse and outdoor growers. This section here talks about major suitability in this region for cannabis, and you'll find that if we mirror it with an eco-regional map like I have here, there's a lot of overlap in places where there are rivers, Arkansas River, Missouri River, also water areas near the Great Lakes and things like this. So this seems to be a really important suitability factor. And if you're not familiar with these regions, they're often referred to as the driftless area, the Western Corn Belt Plains, and the Central Great Plains regions. Overall, though, the suitability is pretty good for cannabis in the sort of Midwestern area. And there's precedent historically for it, of course. And about 300 years of cultivation, there's probably an overlap with the, for example, budworms and spider mites we talked about earlier. And so it's possible that budworms have become actually adapted to. And it's been shown in other research that moths that as larvae develop on one plant, they will seek out that plant aromatically when they become adults to lay eggs if they're female. So this perhaps is true for cannabis already, but we also expect there to be some range changes as climate changes, mostly in places like Missouri, Nebraska, Kansas, Illinois, Wisconsin, and Indiana. This was also really cool. Different organizations, several of them universities, found North American landrace populations in these spaces. So if you live in these areas or can travel to these areas, they might be interesting accession points for environmental resistance to these regions. Researchers have found that in the next 20 years, it's predicted that Kansas, Illinois, Missouri, Indiana, and South Dakota will probably have some level of landrace or feral population loss. But suitability predictors for the environment for cannabis include things like river channels, well-drained soil, disturbed land, that's that rooterol component of cannabis, and ample moisture. And it's been found that there has been local adaptation in places like Nebraska, Kansas, and Colorado, where there are populations that have a European fiber and Chinese landrace component or lineage. Adaptation like this gene called SVB2, which stands for smaller trichomes with variable branches. It regulates trichome production. So changes in this gene has been found. And also what's called MEV kinase, which is important for the production of specifically sesquiterpenes. So that will also affect obviously the terpene and other metabolite profiles in the cannabis in this region. They found that likely occurrence predictors in the Midwest are things like high temperature of the driest quarter, which they pegged as wintertime, low field water capacity, high precipitation in September, and because it was a drought in 2024 when this research was done, they found that September is like the last month with over 0.5 inches of rain. This other really cool research about cannabis origin populations in Eurasia also corroborates that this sort of pink Euro-Siberia, Western Mongolia region, and I would say the central and southern China and Himalayas and North China and East Mongolia regions in green and yellow respectively are also probably sort of related to these populations too. And you might consider trying to find populations that are related to these for maybe like pre-adaptation or exaptation to regions that are like the ones we just talked about in the Midwest and elsewhere. But it's really cool. There are some sort of implications here. On the left, these are different versions of theories about the cannabis sativa gene pool. But I mostly want to say that there is sort of a hemp phenotype in the USA and abroad that could be beneficial, believe it or not, for pests and environmental resistance. We've already seen plains adaptations, and certainly mountain adaptations exist as well. There's a big matching effect between genotype and microbiome. Without the right genes, certain microbes will not associate or will not associate to the same degree, and more research is needed on this topic.
Matthew, excuse me, when you say hemp and land races native to US, are you talking about fiber hemp?
That definitely is included, but also I would include things like oilseed and just regular seed production.
Gotcha.
Yeah. And I would love to say livestock feed too, if that ever becomes a thing. So sort of some cannabis ecozones that are sort of associated with cannabis I have up here are the polar desert and also the tundra, broadleaf forest, and temperate steppes. And this is a little map that I encourage people to come back to and sort of orient towards if you're looking to kind of think about what kinds of populations that you want to grow in your region or from certain regions. Now, this diagram here looks complicated, but is actually very simple. On the left side, we have two sort of selection pressures. On the top, we have basically the environment, which I've identified as like the selection word. But then we also have like fundamental things at like the granular level that influence the development of the plant. So obviously, resource availability, like carbon and nitrogen, will affect gene expression, which of course will affect cell development, and those cells go on to become things like roots and xylem and phloem tissues and leaf and that kind of stuff. So then there's this counter force from the environment and pests and things like this, and those if it's fit, then it'll survive to reproduce, and if it's not fit, then this does not happen. On the right here, we have a simplified version of how plants grow. It's called the growth defense paradigm, and you can look it up, but basically, generally, plants will grow. They'll invest some resources into defense, and genes play a big role in this. But generally speaking, if they can grow strong and fast, they will, especially cannabis. But as soon as it detects certain threats, it has to dump a bunch of resources, finite resources, sucrose and other metabolites, into hormone signaling and defense posture, production of certain chemicals, maybe growing in a stronger, more solid way that is less stringy, so to speak. And the ability to detect and then register the intensity of a biotic threat or an environmental threat is really crucial. Plants are really good at putting just the right amount of resources into this problem, at least in the wild. In domesticated populations, this is maybe a little different. One cool microbe that was found to have a ton of interesting effects, and I also encourage people to check this out more exigently, is the Parabacillus frigoritolerans C114-1. Sorry. And it was found to have a bunch of direct and indirect effects on plants, on cannabis in particular, and these are controlled by genes. The induction of an immune response, neutralizing certain stressors of the abiotic variety, and even the production of certain metabolites. You know, cases of 50 or hundreds of genes related to this. So this is an example of like quantified microbiome research. People talk a lot about bios and the use of sort of having a biodiverse microbiome, but I think that if it's too vague, it's very hard to derive like a real use from those statements. But some microbes are very versatile and some are not, and it's important to know this. How good was it? Well, compared to Serratia plymoutica, which is a like a growth promoting bacteria that is used in other products, it actually increased biomass by 260% compared to control, and stalk diameter and height were increased 96 and 43% respectively, which is a huge change versus control here. So microbes can even augment the physical development of plants, which I think people should make more use of. This is a phylogenetic tree that shows the relatedness of different bacteria specifically in the seed microbiome, the spermome or spermosphere microbiome. An insane number of species, which I've listed here, or different taxa, have been found. Basically, if they're light blue, they were groups that were found most commonly, and if they're more closer to brown or green, then they were found less so. But 813 genera, of which there were thousands of species, is an incredible amount to find in their accessions of cannabis. And what they found was actually there is a relationship between genotype and chemotype that influences the bacteria in the microbiome and different levels of domestication. I'm not going to go over all of these examples here, but I really encourage people to screenshot or, again, to come back to this video and look at all these examples. I wanted to highlight these for people to have a reference, and I also want to bring up this point that it's extremely difficult to compare microbial diversity and effects without controlling for things like phenotype and genotype, environment, even the growth stage versus a propagated cutting versus a mature plant, or even in between. There's huge differences in the microbiome that change over time and space, just like the pests that we have to deal with. So a few examples here, richness and uniformity increase with maturity, and it seemed like microbes would often enter into the plant as endophytes. And this is all mostly to do with endophytic bacteria in particular in this research report. Fungi are a little bit different, we'll get into that. But a lot of them moved up from the substrate into the plant tissues. The core microbiota across domestication levels from landrace to inline bred to feral were Bacillus, Pilomonas, Ralstonia, which is sometimes considered a pathogenic genus of bacteria, Staphylococcus, and Pseudomonas, which has good guys and bad guys, so to speak. Fungal endophytes in a hydroponic greenhouse, so not a soil substrate area, had endophytes that were associated and not associated with that substrate. That substrate, I believe, being, if I remember correctly, cocoa coir or rock wool. But you can check out the research report down here, which I've cited. Some of these fungi, like Fusarium and Aspergillus, are known to be pathogens, or often are pathogens. Some species are more benign, but you're not really going to know in your substrate unless you do some testing. But it was interesting that all tissues found two penicillium species that produce antibiotics. You know of penicillin, of course, but also citrinin, which has implications for maybe biocontrol effects against other bacteria. that are pathogenic, and other microbes too. But also, interestingly, Rhizophagus irregularis, which is an arbuscular mycorrhiza, was found in all tissues, not just roots, leaves, stem tissues, and flower tissue as well. It's been found to have effects in many plants on nutrient and growth stimulation, but also, in our case, cannabinoid yield, which is interesting to me. And there are some other facts here, but the main point is that endophytes and other microbiota, they can be beneficial, they can be neutral, they can be detrimental, and that context can even change over time. I wanted to go through these tables really quickly just as a reference for people if you want to look for some of these bacteria that they found in products or other references. We have here feral and wild cannabis versus indoor cannabis. And you can see there's a difference in the seeds. There's a difference in the leaves and petioles between the more less domesticated and the more domesticated populations and their growth context. Here we have on the right crowns and stem bacteria, and on the left we have examples of like penicillium, and in F here, fusarium moving up through the xylem channels, so the water channels of the plant, which of course connect to the roots, right? And then we have some root bacteria here, again, from the two different populations. And on the right, we have more microbes that are, again, sort of moving up through the xylem vessel elements. These are spores specifically. And then a really cool table that showed different species, effects on the plant or pathogen that they had, and their mode of action, which I think is really important. When people are telling you that you should spend your hard-earned money or your resources or your time on using certain products or having certain kinds of processes, you should ask them why and how they know that that's true, and ask for some data, because these people respected your time enough to actually try to quantify that. But the gist of it is that there was a lot of strong inhibition. Again, probably hard to see. This presentation is meant to be seen on a screen. So if you're expanding this out, you might not see this, but take a look at this table when you get a chance in detail. There are a lot of fungi that were found to be inhibited pretty strongly, up to 93% or more in some cases. Even something like trichoderma, which is oftentimes, especially this one, used as a biocontrol of fungi. It was found to be an endophyte that had effects on the height, dry biomass, and root density of cannabis plants, which is pretty incredible again. Some people have been dealing with things like leaf blight or leaf spot fungi. There are many species, Septoria, Alternaria, Bipolaris, and they all have very similar symptoms, and they become a huge problem for a lot of growers, both commercially and home grow. But they found a Bipolaris CSL1, and here this is a tree of its relationship with other Bipolarises, typically a pathogen, but a beneficial endophyte, isolated from cannabis, increased gibberellin and indole production, which are two growth hormones in plants. It also inhibited jasmonic acid in the host in cannabis, and also chelates iron, making it more bioavailable. So they put this in rice, and you can see a difference between control and the CSL1 inoculated rice plants. Pretty neat. This diagram here shows different stages by which these PlayStation symbol microbes, these are meant to be sort of euphemisms for different microbes and their movement. Basically, you can see here like seeds, they only get the X and square microbes, whereas you might find a larger amount of microbes in different tissues here. And then to briefly close out, I wanted to talk about this complicated mess, which is basically the way that data is probably going to be processed in the future. And it'll influence things like our biosecurity decisions at the micro and macro stage. And I think that this will become way easier for people to get access to on the individual level. It'll become democratized. And I think this global forecasting for things like those nasty strains of pests I talked about, but also cool microbes that we're interested in, this information become much more accessible for people. But it's still kind of in its infancy in a way. But some of the cool things it'll allow us to do is simulate pest spread and major factors. What we as growers have to consider, and what extension agents and other people will influence different products, and hopefully safer, more sustainable products to be used, because we have data to back up their effect. And so in the next 10 years, I expect that this will be also true for cannabis. And that's it. Thank you for having me.
Amazing, Matthew. Thank you so much. I have a question for you about everswarm. Can you explain what that concept is?
Yeah, so it's like having it's just having an acknowledgement that the pests that we deal with and also the plants are constantly evolving. And so if you're not considering that your options, your strategic plans could change, their efficacy can change, then I think that you'll lose at the speed of their evolution.
And eco-maxing?
Yeah, so eco-maxing, that's just me being a little bit cute. Basically, you want to maximize the ecological effects that are positive for you, and then leverage the disadvantages as much as possible on the pests.
Got it. Understood. Okay. There was a question in the chat, somebody asking about nematodes, getting very specific. Any recommendations for nematodes for thrips?
Over thrips, good. Okay, so some people do use nematodes for thrips, even in the foliage in some crops, so this is a niche use case. But for in the soil and the substrate, it depends on what you're controlling. So a lot of people are using them for thrips, but also for fungus gnats or shore flies. The thrips, they have um, a form that doesn't move. It'll fall into the substrate, and then you can use nematodes to kill it. I like to use Steinernema feltiae. It also goes after things like fungus gnat larvae too. And it's probably my favorite use for a biocontrol for specifically fungus gnats. That's why my mind immediately went to them as sort of a cross pest you can kill two birds with one stone with.
Very cool. Love it. Another question. Any recommendations for rearing lactis mites?
Lactis.
Lactis mites. I don't know what that is.
Oh, I'm not sure either.
Okay.
It's possible they mean there are like cheese mites, but I think they have a different genus name, and they're used as a feeder mite if I'm interpreting that well. But if there's another question, if they can clarify, I'm happy to go
Yeah.
over it.
Yeah. I do have another question for you too. In the beginning of your presentation, you were showing how the flow of trade carries these pests and disease across continents. And if you look at that on a very micro level, the flow of things in and out of your facility controls your flow of pests and disease into your facility, basically, right? I mean, what you bring into your facility is what you're going to have to deal with. If you don't bring anything into your facility, you don't really have, you don't have any pests and disease to deal with. I mean, necessarily. I mean, there are some things that are going to be in the soil that can grow out of control and get out of proportion. But yeah, I was wondering if you could speak about that a bit about preventing the disease and pests in the first place from taking hold.
It's a salient point. Right. So on the micro scale, right, it's scale agnostic, this everswarm concept, this idea that like things are constantly changing, things are moving in and out. So on the macro scale, you have this effect that you should be informed by. But then on the micro scale, I agree. A lot of times, because of the way that bios work, the way that chemicals work, and I'm a proponent for using safe, effective products. Um, but a lot of times I think the it's like that quote, it's an interesting game. The best way to play is to not play, right?
Hmm.
To not play with the pests, to not give them the platform to evolve under which. So a lot of times I like to use physical barriers like having indoor grow tents or having growing in your room and having like really good controls for that, especially for home growers that don't have a lot of resources to deal with. But it's also true commercially, honestly.
Yeah. Yeah, very cool. Another question, how do you deal with fungus gnats in home grow hydroponics? Oof, that's a toughie.
Yeah, so I would say that you should always identify, how are these pests, like you just said, right, very, very adroitly, how are they getting in? If you don't know that, you won't be able to solve your problem, in a holistic way. But one of the things that works against them, like I mentioned, is those Stannernema feltiae. SF nematodes, very effective, but I would say trying to make sure that those pests don't have an easy into your space is really crucial. And there are ways that can complicate this. You know, you might have like a leaking, if it's in your home grow, or if it's like around your house, your property, if you have like a lot of waterworks, or if you have something that might be leaking, or a pipe or something like this outdoors, that can cause like a oversaturated substrate that fungus gnats can kind of enjoy sometimes. But yeah, it really is going to be contextual in that way. But that's one thing you can use. You can also inoculate the substrate with like some Beauveria or Metarhizium fungi, which will also colonize the fungus gnats. But I like the nematodes the best.
Now, the best way is just not to get them in the first place.
Of course, right.
Awesome. Well, let's see. One more question here. Can you please repeat what you're using for? Oh, can you please repeat what you're using for gnats instead of the nematodes? One more
Oh
time.
yeah, sorry. So Beauveria or Beauveria is a type of fungus, Beauveria bassiana. If you type that in, you'll probably find the sorts of products that are like this. And Metarhizium, which is another similar kind of fungus. And these guys you can apply to the foliage or to the substrate, and they will colonize most insects. Rice root aphids are another thing I like to use against with these.
Very nice. Excellent, Matthew. Well, that's all the time that we have today. Matthew Gates of Zenthanol Consulting and Avitas Global, thank you so much for your time today.
Happy to be here. I look forward to our mutual success.
Thank you, everyone. Thanks for joining us. I'm Michelle Navedo. Have a great weekend, everyone. Bye-bye.